Good day, ladies and gentlemen, and welcome to the SNN.Network Summer Virtual Event. Our next presenting company is GeoVax Labs. It is now my pleasure to turn the floor over to your host, David Dodd, CEO of GeoVax Labs. Sir, the floor is yours.
Thank you. Thank you so much. It's a pleasure to have this opportunity to tell everyone about GeoVax. I apologize. Just need to get the right slide there. All right, GeoVax is a clinical stage company. We develop vaccines and cancer immunotherapies for a number of infectious diseases. Our lead program is our universal coronavirus vaccine, which we'll talk about in this presentation, as well as our cancer immunotherapy program, which we're advancing towards clinical development. We're well capitalized, having no debt and financed through 2023. Last September, we were successful in recapitalizing the company, raising significant capital, and listing on NASDAQ. This was followed by additional announcements about our progress during fourth quarter. During first quarter of this year, we did additional capital development and now have approximately $20 million on the balance sheet. We have no debt, a very clean balance sheet. We're financed through 2023. However, if you were to look at our current burn rate, we'd be financed for almost 5 years, but we plan on continuing to invest to accelerate our programs forward. As I mentioned, our primary program is our universal coronavirus program, which we're focused on developing a vaccine in a single dose requiring minimal to no refrigeration that is durable and simple and able to be distributed. That's followed by our cancer immunotherapy program. Our other programs are being driven, especially those that are our Lassa, our Ebola, Marburg, Sudan, the hemorrhagic fevers. Those programs are being advanced through the support and non-dilutive funding through either the U.S. Army or NIH. All of them are progressing through non-human primate in preparation of clinical trials. Our technology is one that allows us to take a different approach than what other vaccine developers are doing. We're able to encode or to incorporate multiple components of a viral structure into our vector carrier. This is unlike other technologies, for instance, with mRNA or even the adenoviruses, that respectively, that's what is used by Pfizer and Moderna for their COVID-19 vaccines, and also Johnson & Johnson, AstraZeneca, utilizing the adenovirus vaccines. In those instances, one is only able to incorporate one protein at a time for the virus. Ours is able to incorporate multiple proteins. We typically will incorporate a lead protein with certain structural proteins, and in doing that, it gives us the ability to generate a single-dose performance vaccine because we're generating not only antibody immunity, but also cellular or T-cell immunity, that which is critical for memory and durability. All of us are aware of the continued emergence of the variants related to COVID-19. Most of us have heard almost every day throughout the day about the Delta variant, but what we don't know in some instances is that there are now at least 4 future variants that are already out there impacting people. Recently, through work both at Fred Hutchinson Center in Seattle and Emory Vaccine Center in Atlanta, there was a paper release that acknowledged the importance of being able to adjust more than the simple S protein or the spike protein to be able to give the necessary coverage, to be able to provide the prevention and the benefit that we're trying to seek for people worldwide. As an example, WHO recently laid out that beyond the Delta, we're already talking about four additional variants, the Eta, the Iota, the Kappa, and the Lambda. So this is not going away. It's a major issue, and so much so that we've already seen additional calls for proposals for funding from both NIH as well as CEPI. looking for technologies that can provide in a single dose a much broader variant coverage than simply the alpha or the initial protein that has been focused on. This has been the approach and is the approach of GeoVax. Our lead candidate is known as CMO2. We are currently completing various animal results. In fact, tomorrow in an international conference, the European Society for Medicine our CSO will be presenting data behind our program, and then that will demonstrate that through both hamsters as well as guinea pigs, et cetera, that we've been able to provide both antibody as well as cellular immunity, and we do this by incorporating both the matrix and the envelope protein in conjunction with the spike protein. Based upon these results, which we've already been sharing with the NIH, we've already submitted a much larger application to the NIH for funding that will enable us to accelerate to the clinic. So we refer to this as our GOVX program. As I mentioned earlier this year, we received our initial funding in support of our concept, and which we've acted upon, and that was from NIH, which enabled us to formulate and design, or take this product we'd already designed and constructed into initial testing towards a single-dose vaccine against multiple COVID strains. That is the purpose behind which we are working right now. The technology we use, however, is not new. It is not unproven. It's been well validated in the past. There are vaccines on the market that use MVA. And in fact, in our own work previously, we've shown 100% protection in a single dose without the need for any additional components or adjuvants through the Zaire Ebola, in the monkey model. And other animal models, we've shown similar strong performance in a single dose with the Lassa, the Sudan, and the Marburg. In fact, these data have been sufficiently impressed that the U.S. Army is now funding the Lassa program through non-human primates in the preparation of CGMP material to be ready to move into the clinic. And the NIH, through its preclinical services, is currently testing our Sudan Ebola and our Marburg, also through non-human primates, to be prepared to move into clinical development. We believe that based upon delivering promising results as we have already with Zaire Ebola, that we can move forward and become part of the Strategic National Stockpile Program. In a similar manner, the same technology has previously shown 100% protection in a single dose against the Zika virus. That was one in animal testing also. We've opened up discussions with parties in South America about moving this forward into the clinic, mainly because the threat of Zika remains a major threat in the Southern Hemisphere, specifically in South America as well as in parts of Africa. In HIV, which was the basis upon which the company was founded, although there is not today as much of a focus and support to move vaccines forward for the prevention of HIV, we continue to have completed through a large phase 2A program. We are scheduled for an additional phase 1 program evaluating a novel booster, and also we are actively involved right now in a program at University of California, San Francisco, looking for a functional cure. So we continue to support that, but only upon receipt of non-dilutive funding. One of the most exciting areas in which we've entered in recent time has been that of cancer immunotherapy. And in that regard, our approach has centered on constructing a tumor-associated antigen vaccine and combining that with an immune checkpoint inhibitor to enhance the utility and the performance of the checkpoint inhibitor. Our initial focus has been with the mucin 1 or the MOC1 tumor-associated antigen. That is a tumor-associated antigen that is widely expressed in many different cancers, so there are numerous targets. It also has been in clinical trials, is currently in clinical trials, so it gives us a much faster entry into that arena. We have recently completed and presented at various conferences the results of some animal work in a therapeutic experiment model. We took We took genetically modified or transgenic mice that all had been exposed to a human tumor. One cohort that was untreated continued to develop the tumor. Other cohorts, which was either just our vaccine or just a checkpoint inhibitor or a peptide, it continued to grow, albeit at a lesser manner than the untreated group. But the group that combined our vaccine with the peptide and the checkpoint inhibitor, it showed a stabilization at day 30 and then a decline. And then to further evaluate it, we then tested in a recurrence model, which was really in a genetic mouse, a prevention model. And in that regard, these were tumor-free mice, presented with a human tumor. The cohort that had no treatment, 100% developed the tumor. The cohort that received our vaccine, MACH-1 vaccine plus the peptide, had 100% protection. And this is the basis in terms of the therapeutic model that we are preparing to move forward with anticipation of filing an IND in the second half of next year and moving into the clinic. We continue to strengthen our intellectual property and have recently made some announcements to that regard, and we'll continue to do so to ensure that our technology is well protected. So in summary, and then I'll turn to some questions that have come in, our primary focus is on our universal coronavirus vaccine program, which, as I mentioned, we're looking for various niches, as well as our product can also be used as a booster to further enhance and provide additional protection against emerging variants. It could be utilized in conjunction with mRNA or in conjunction with the adenovirus or by itself. And then our other primary focus is in our immuno-oncology. So with that, let me turn to see some of the questions. So there's a question here. Is there an emergency authorization that GOVX has or will apply for to get these vaccines approved? The answer is yes. We'll pursue that avenue, and we would expect that we would receive that as we're prepared. We've already had discussions with entities within the federal government about that. We believe that our advantage, there's a question here about what advantages we see over the existing vaccines for COVID-19. Specifically, it's all based upon our ability to elicit both not only antibody immunity, but also the cellular immunity, given a broader coverage, and by doing that, being able to encompass what may be emerging variants before they emerge. So with that, I don't see any other additional questions, so I just want to thank everybody for your time and for your interest, and I look forward to all of us having a great conference experience thanks to SNN.Network. Thank you.
Thank you, ladies and gentlemen. That does conclude the GeoVax Labs presentation. Thank you for joining us. You may disconnect your lines at this time, and have a wonderful day.